Neil Amin with Sculpta Bio. Welcome, Neil.
Hello. All right, let's see if my voice still works. It kind of does. I had a little bit of a cold before, but then I woke up yesterday and just shot voice. So I apologize if I was not very social yesterday. I wish I could have, but wanted to save my voice for today. So clickers here. So first off, it's exciting to be here. Exciting to hear about all the other companies and just the great energy around neurotranslation. So my name's Neil. I'm a physician and a scientist, and I am excited to tell you about our company, Sculpta. We are sculpting the future of human brain health for patients with neurodegeneration, as well as for the aging population. So my background, as I mentioned, I'm a physician, a scientist, MD, PhD. I did my residency and postdoc at Stanford, became clinical assistant professor there, and for a long time, really loved what I did in academia. Published lots of papers just in the last year, 2 papers in Nature, a first author paper in Science, and another first author paper in Cell Stem Cell. And probably from the outside, it seemed like that's what I'd be doing for my whole career. But when I first became a physician scientist, or wanted to become a physician scientist, I really believed in the idea of taking a really deep dive in basic mechanisms of biology and taking them to patients. And I lost faith that I could do that in my academic training path, and took the big leap 1 year ago, quitting my job completely, to doing something crazy of launching a company. I'm a first-time founder. I'm a solo founder. I never worked in biotech. And I was raising in the worst biotech market that we've seen. So clearly I'm a masochist. But really, I was just stunned to see how many people really believed in our vision, wanted to support neurotranslation. And we raised 2.6 million, oversubscribed round in a pre-seed from some really phenomenal investors. And that was just around 6 months ago. Now we have this fantastic team based mainly in San Francisco, but also with folks in Croatia and the Netherlands as well. And so I don't really need to tell this crowd about the importance of brain of drugs, developing new drugs for the brain. But I do feel like there's a lot of excitement right now because there's growing appreciation of the population's only getting older, and neurodegeneration rates are only getting higher. The FDA has signaled that with all the availability of biomarkers, that they're willing to approve drugs based on these biomarkers, such as the approval of tofersen based upon biomarkers just alone. And I do think there's excitement with actually generating therapies for Alzheimer's disease, despite their shortcomings, that now there's opportunities to develop additional therapeutics on top of the ones already approved. So here at Sculpta, we're very interested in neurodegeneration and the aging brain, and really finding therapeutic strategies that allow us to expand the opportunities of drug pathways and pathophysiology beyond what biotech and pharma are currently doing. And so what we know about disease pathophysiology and neurodegeneration is that over time, your brain accumulates different proteins. These signal and trigger lots of inflammation, and this will signal to neurons and eventually cause their death. And this really causes an immense amount of brain shrinkage over the lifespan. Protein aggregates can look like amyloid in Alzheimer's, synuclein in Parkinson's, or even just in aging. There's a protein called lipofuscin that just develops builds up over time. And recently there anti-amyloid therapies have been approved that really effectively clear amyloid from the brain, but unfortunately the clinical benefit was rather modest. And despite the billions of dollars, truly billions of dollars spent on amyloid and tau over really decades, there's just limited clinical benefit and really a need for new directions in therapeutic avenues of really treating the disease. Also, the downsides of these antibody therapies is that there's increased risk of brain inflammation actually caused by the therapy itself, and also very frequent dosing. So when we looked at how can we really innovate in drug discovery through novel target pathways, it became really clear to us that the biggest drivers of Alzheimer's risk from really high confidence genetic genome-wide association studies are really landing in microglia. So these these genes associated with Alzheimer's are really highly expressed in microglia. Some of them really specifically expressed in microglia. So what are these cell types? Oops, too far. There we go. So these are the brain immune cells. So even though there's been, I don't even know how many drugs that modulate the peripheral immune system, there have been no drugs ever approved that specifically target the brain immune system. I think it's a completely missed opportunity because these cells do an incredible amount. First of all, they clear protein aggregates from the brain. They modulate inflammation directly, and they also directly signal to neurons to protect them from damage. So with all of these source of evidence pointing to microglial function, we really believe this is an exciting area of therapeutic development. So when I started the company, we really felt like the most powerful genetic tool that we have to get into new targets are oligonucleotide therapeutics. And it was brought up before, and it's great you guys already have a little bit of background on why they're exciting. But what inspired me and many other people is that back in 2016, when I was a graduate student, the drug Spinraza got approved for spinal muscular atrophy. This is a disease that's fatal for children. There's no treatment for it. And all of a sudden, you didn't need a stats background to see in that trial that most of the children were now surviving. It was remarkable and still is remarkable to this day that a new treatment paradigm could work so remarkably well. I think what makes this drug category especially powerful is that it is all the features of a futuristic drug. You can change the sequence, you get a new drug that targets a new part of the genome or transcriptome, but it has the same uptake into cells almost all the time. And this is now not new. I mean, there's now 20 approvals. I think the next decade we'll see that of all the genetic medicines, CRISPR, AAVs, that oligonucleotides are really the most clinically translatable. So where we come in is really expanding the use of this drug category to a wider range of targets and with different mechanisms, using our knowledge and of basic biology as well as our technology around single-cell genomics. So our approach gets away from this paradigm of just trying to knock stuff down or increase levels, like say knock stuff down with CRISPR or increase levels with AAVs. We're actually doing what we call protein sculpting. This is where we actually change the structure and function of proteins in the brain. So how is this possible? This is through modulating alternative splicing with these antisense oligos. We can do this by tapping into existing differences and splicing that occur in the brain, or even create de novo variants. And just to give an example of how we can use this, is there are certain proteins that sit on the cell surface, a receptor with a transmembrane domain. We can actually skip the code for that transmembrane domain, and now this receptor is, instead of sitting on the cell surface signaling disease pathways, can actually be secreted, still binds its ligand, but no longer signals. These are the kinds of pathways that you can never imagine doing with a small molecule and are really specific to this pathway. So what gives us a very competitive edge is our technological background that combines GWAS data with really advanced, the most advanced single-cell transcriptomics in splicing. So you might have seen around the news, we're kind of in the single-cell genomics era, but 99.9% of that data is unusable for splicing discovery because it doesn't capture the full length of genes. We have built the largest database of single-cell RNA splicing data by accessing roughly $30 million of data that's never been before analyzed in this way. This work is partially sponsored by AWS and Google. And we built the first human splicing atlas. We want to expand this to Alzheimer's, Parkinson's, and ALS because never before had been generated. This single-cell splicing data has never been generated from any of these disorders ever. It's a really big opportunity for us to discover new targets. So from our platform, we already identified 2 targets that pharma is not going after, but we have built a very compelling case for it based upon our GWAS studies and our single-cell splicing analysis. They both target microglial function in the brain for Alzheimer's with very high evidence through GWAS studies in Alzheimer's. We have in vitro evidence as well as a pathway to clinical trials because we actually have we're actually modulating a validated CSF biomarker that we didn't have to develop ourselves, but is really there that we can use to see if our drug's really affecting our pathway both in animal models as well as in humans. So I mentioned, we raised 2.6 million. It's really just enough to get us off the ground. We have very big ambitions of what we want to do. Our platform is really in the middle stages of development. We have really new technologies to actually capture single cell splicing from these disorders at a scale that other people have not, through a new sequencing approach called BobSeq. That'll be we'll have a limited release early next year. And our two assets have been identified for our platform. We're moving to iPSC models. So my maybe around 7 years at Stanford was all around developing the most advanced human brain organoid models that anyone's seen, and we've had tons of papers on this. And really, this gets us out of the kind of just the stuck place of trying to overly rely on rodent models for modeling human disease. One really important point here is that our second target doesn't exist in rodents. This happens all the time where human genetics lands in a gene or a gene pathway that just doesn't exist. And this is actually true with the drug Spinraza. SMN2 does not exist in rodents. If we limit ourselves to only gene targets that exist in rodents, we'd be much worse off. And so we are not shy about this, that we want to highly leverage our iPSC models and then move into non-human primates to advance these candidates. And so our goal is to raise a $10 million seed round to advance our platform and assets next year. And really importantly, it's been super exciting being here because I've seen how much Chris is he said today's his favorite day, but it's clearly the case that he's been working on this every other day of the year to build a community of people. Like I'm in the Bay Area, there's a great ecosystem, and that's really what it takes, is an ecosystem of scientists, biotech entrepreneurs, clinicians, really everybody coming together. And it's very clear that he's brought everyone, here in the same way. And so what we're looking for is obviously always fundraising, and we're very excited to bring in new folks to into our company. But also, we have an exciting platform that can be applied across many different areas. We're really interested in generating new data sets from any kinds of clinical samples that you guys might have. I know there's a lot of also basic research going on in this area that can lead to new drug targets. We already have one partnership with an academic center, helping them advance their target. That's what we're all about, is expanding the pool of target opportunities for patients. So thank you all. Appreciate it. And I'll take questions.
Okay, great presentation. Thank you for being here. I'm sorry you don't feel good. You couldn't tell during the presentation. Couldn't tell.
My voice just doesn't feel great.
So question for you. You mentioned that amyloid and tau aren't moving the needle far enough, so you're looking at a separate mechanism. But you didn't really talk about what biomarkers you're using to actually show the activation. And while you reference the FDA biomarker approval pathologies, they're actually not in AD with the exception of amyloid. And even then, it's just accelerated. So question for you, I don't know if the CSF biomarker you mentioned is confidential, or if you are thinking about a difference between biomarkers for engagement, target engagement, versus tracking response to therapy.
Great questions. And because our innovation is in new target discovery, we can't say much about exactly what our targets are or the biomarkers which people will be able to figure out. But our approach is really considering this way, that we want to have both a biomarker for target engagement, but really the real money, so to speak, of showing efficacies in validated biomarkers that have been well accepted as actually being related to Alzheimer's pathology. So that's what we need. I mean, that's the bar for really having a clinical trial. So that's what we're going for.
So you're really looking at the existing biomarkers that are validated with longitudinal cognitive assessment data.
Yes, that's right.
Okay.
Yeah.
Why do we need that?
Why do we need biomarkers?
That we already have.
Why do you need
No, I just So it's the same question you just asked. So we have tons of biomarkers, especially in the CSF, that are already validated or approved in clinical use. So I'm just what are you doing beyond that?
Yeah, I mean, right now, what I think is exciting is that there are now biomarkers that we don't necessarily need to develop both new assets and new biomarkers to get it. That's a lot to ask for. What we are actually developing is, and this is more on our platform side that I mentioned, is developing the first splicing biomarkers of disease. So we're really interested in collecting CSF, and you can actually collect some microglial cells in CSF and routinely in CSF. No one's ever used this for tracking any splicing drug that's ever been developed. I think it's a real opportunity for us with our single cell splicing analysis to actually develop wholly new types of biomarkers, actually great for actually target engagement. You can imagine if you're injecting a splice switching oligo, as we want to do, that you don't know if it's splice switching in living cells. And the opportunity to actually get cells from CSF is really a huge opportunity, and people just haven't done it before.
So just to clarify, to understand what the common nature of a lot of what your target discovery might look like, are these generally transmembrane molecules with internal signaling that themselves already go through endogenously alternative splicing? So there's up and down regulation and alternative splicing, and you're trying to nudge it toward that splicing that shuts down that signaling so that it stays in that state where the intracellular portion is not active.
Yeah, this is a great question. So the background here is that 90% of genes have alternative splicing, and when you really look at the data, it's like there's a little bit of noise everywhere. So there's a little bit of alternative splicing going on all the time. So it's a little hard to define like what is happening or not. For one of our targets, roughly 10% happens at baseline. It's been like we can see it in actually primary human microglial samples and in IPS models, and everywhere we look, it's around 10%. And then for other target, it's actually that one, our second lead target is at 50%. We have other targets where we have to be very selective where we go around, but some are actually completely novel in that it's really low, extremely low levels. I think what's nice about if there is some baseline splicing, it actually gives us some indication on the biological side that it's actually easier to modulate those. If something's really zero, if there's something about the cellular machinery that's really trying to prevent anything from skipping that. The other interesting thing is that from evolutionary standpoint, there's actually a lot of reason that we can actually do this across almost every gene with multiple exons, because the exon shuffling hypothesis of evolution that most exons contain a domain of a protein. So it's not like there's randomly scattered, the sequence is randomly scattered. What that means is when we skip a exon, we're actually just removing one domain, the rest of the protein folds normally, and that allows really wide space for us to explore new mechanisms.
Yeah, just stay away from the bio weapon.
Oh,
right.
I hope not.
If you're tweaking what evolution is keeping you from ever alternatively splicing.
Yeah. Well, in the genetic medicine space, that's what we do.
Yes.
We play with genetics,
dual purpose.
But absolutely, yeah, we definitely want to be safe. Yeah, absolutely. Yeah.
Great job. What would the uses of funds be in the context of this this competition and in your current role?
Yes, so we're as you can tell, we're a very early stage company. We've just been close around 6 months ago. That money really makes a big difference for us. First, I'll just say this competition makes a big difference for us. It's all about just getting out there and really appreciate that already. But first, the money really helps us push how much we can do with our animal models. I think that's really that last bit that's elevates our impact and gets attention for us. And so that money really will be directed either towards, we already have quotes for non-human primate studies, will actually get us through our initial pilot testing there. And that would be really huge for us. Thanks. Thank you.